And people do talk about the pricing, and if you go back to my first podcast, I had mentioned about the cost of cell therapy.
Right. Yeah.
Now, I'm a little bit older, a year later, a little bit more educated, and they're still really expensive. However, if you look at the health economics, and you look at how much the healthcare system would be paying,
right,
over the course of someone's life, of that chronic care.
Yes.
And so the burden on the community, the family, the healthcare system, and if you do the math, it actually is a good investment to treat that person, cure that person.
Yes.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe.
Hi, and welcome to BioTalk Unzipped, where we unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin, a life science professional from Kansas City. And today we are at AAPS NBC in Boston. And today we are joined by the AAPS President, Russ Weiner.
Hey,
good to see you, man.
Thanks for having me.
I'm glad you're here. Yeah.
It's going to be fun. Go for it, man.
Yes. All right. Well, this is your first official, conference as AAPS President.
Correct.
So how is it different than your other AAPS experiences, and how are you soaking it all in?
The difference is you can't walk down the hall without somebody stopping you and talking to you. So a lot more time is in my schedule to get from point A to point B.
You need to.
A little bit what I call AAPS groupies.
Nice.
It'll be worse at the national meeting because that's when the students come out and they all want to meet the president.
Of course.
But aside from that, I still get to attend sessions. I still get to have poster sessions where I get the individuals that I've selected from the program, which I think are the top 5 in my interest. We can do those. Other board members do that as well.
Nice.
And then you just get to enjoy yourself, right?
Good.
I love to get on that stage and just go a little crazy.
And we love it when you do. Never a dull moment.
Nope, hope not.
No way. And I know your passion, is mentoring people, and especially younger folks. And this one may be a little bit more special because you have your son Jacob with you.
Yep.
And so he's able to see a little bit from your perspective, your lens, and getting to see that experience. How has that been for him, and how fun has it had to have him around for this?
Well, for me, it's the proud daddy moment as I'm walking with him, and then when people look at his last name, they go, I think that's his son.
Yeah.
Maybe. But he's having a ball. He's not a shy kid, as you've seen, and so he's, out there talking as if, he's been out in the field for 10 years. It was quite remarkable.
Nice.
Even last night at dinner, he's sitting around with, senior people in the organization, and he doesn't back down. He sticks to his guns, and it's quite refreshing to see that. I haven't seen that, with him. Around a round table with, conversations coming in every direction. But for me, it's so fun to just hang out, and we generally sit in the same sessions so I can explain to him what some of the acronyms are. Right? And that's where he gets lost a little bit, but you have to be in the field. And I think just seeing all the different things here has just made him super excited to graduate.
Yeah, no, that's great. That's great. Well, I'm glad when you threw him in the deep end, he swam to the edge. That's great.
He's not a shy kid.
Good. How is this, first conference of the year setting things up for Summer Scientific Forum in July and PharmSci in November? So
this starts the meeting season, as you know, and the focus of this meeting is varied, right? It's biotech, but it's varied, a lot of different areas. The Summer Scientific Forum sets you up for more focused bioanalytical or pharmaceutical type presentations. It's a much smaller meeting, more of a, I wouldn't go say it's a Gordon conference, but that's the size and that's kind of the feel you get. And so this allows us to see, particularly for PharmSci 360, what rooms are packed with people on certain topics.
Sure.
Right? We have not done the final programming for that meeting, and the goal is to see, what either is super hot or needs a second session. That session really should have gone 2 hours, but we're not going to run a 2-hour symposium.
Sure.
And so understanding how many people are in that room, and then when we get to sci-fi,
Farm side.
Farm Sci 360, we have a better feel for how we should stage, some of those rooms. So it does, and the vendors will decide, where they want to go, and I think they see here the tremendous amount of one-on-one interactions, and the same thing happens at the Summer Scientific Forum. It's that small face-to-face, have a drink, talk business, and it allows the vendors to come and see a whole varied number of clients versus having to visit a dozen clients. You can get them all one shot.
Nice. I like that. Well, it's a great approach to set things up for even more success the rest of the year.
Yep.
That's great. That's great. To shift a little bit, you've recently made a change. You're with Lexion now, and back in the rare disease space. How is that hitting, and how are things kind of getting started back into a space that touches your heart?
Well, first of all, it absolutely touches my heart. As you may or may not know, I got into science because my mother passed from multiple sclerosis. And we have numerous friends, pick your rare disease. And we have one right now who has rare cancer, and is an 18, 19-year-old kid.
That's tough.
And he is pounding through it. But young people can pound through it, whereas the older people have a little bit more difficulty. But the beauty here is you get to work on the most interesting, life-changing treatments for diseases that go misdiagnosed for many years. So the big issue that I've learned, aside from struggling with understanding the many different rare diseases and the different targets, having come from more of an oncology background, where I understood all those targets, I understood all the genes that they were going after. Here, not so much. And then layer on top of that the fact that we have different modalities, and it's not just a single modality. So I started with antibodies and small molecules, then we went to fusion proteins, and now we have antisense, we have siRNA, we have siRNA attached to antibodies, we have ADCs where we put drugs on antibodies, we have trispecifics, bispecifics, T-cell engagers. So layer on top the different diseases, the different mechanisms, and the different modalities. The learning curve is much higher than I had expected. I would have expected I'm about 6 months in, that I would have a good handle on the portfolio. Well, that's not a true statement, and it'll take me a while. But the beauty is we saw a patient come through who has myasthenia gravis. And individuals, with that disease, they often, can't see well, they can't walk well, they can't swallow well, and this woman just walked across the stage and spoke to us for an hour. And that is like, keep those tears inside, boys. Right? So while it is extremely difficult, it's a level of reward that you can't really compare. And the closest that it would have come to was my time at Bill and Melinda Gates Medical Research Institute, again, working with people who have terrible diseases that people may or may not care about, or they're so far away that no one thinks about them. And yet, each one of those diseases, whether it's TB, malaria, Shigella, you pick it, they're all just one flight away. And all you need is one person with TB to come over and sits on a plane, and it'll just spread like wildfire because we don't have vaccines for TB. So you can't say it's not in my backyard when that backyard will be your backyard if you're on that plane.
Indeed.
So it's definitely a challenge of getting these patients, and that's actually the hardest part. Most rare diseases are poorly understood, and if you're a community physician, you may have never seen that disease in your entire career. And there are 10,000 different rare diseases. So how does these family physicians, people that are just out there, how do they learn about this? And so what we do know is that patients take between 4 and 7 years to get a proper diagnosis. And you can imagine, for a not rare disease, a regular disease, you go and you have cancer, they believe you have cancer, they take a tumor biopsy, and then you have to wait like a week. And a week is a long time. Now imagine it's 4 years, and the stress and the the amount of pressure that puts on the family, the child, the healthcare system as they go through test after test after test. And so things that are out there now are what they call AI diagnostics or software as a medical device, whereby we can tap into electronic medical records. We can take tests that may have happened at 6 different community hospitals, or maybe they went to the big city and they got some tests. Can we aggregate all that information and then use AI to say, wait, this person has something, and it's of one of these 2, or it's this. And so changing that 4 to 7 year paradigm down to quickly.
Yes.
Right? Because some of these people, these diseases, you don't live very long after they've really set in. And 4 years of it setting in hard, even when you find out what it is, it could be too late.
Right.
So that's the biggest challenge, is finding what that disease is.
Yeah. Early indication, early diagnosis is dramatically life-changing. And life-saving in that situation. So no, I appreciate that, bringing this human touch and this humanity into what we do, because some people from the outside looking in, it's this business and it's expensive and what are they doing to it? Those types of things. No, there's people who are, as you mentioned last time we talked, they're sacrificing their time and their lives, and they're working hard and working late to bring these medicines, to the market, that will extend lives, that will save lives, that will make you feel better. And got to remember that, it's important.
And people do talk about the pricing, and if you go back to my first podcast, I had mentioned about the cost of cell therapy.
Right. Yeah.
Now, I'm a little bit older, a year later, a little bit more educated, and they're still really expensive. However, if you look at the health economics, and you look at how much the healthcare system would be paying over the course of someone's life, of that chronic care, and so the burden on the community, the family, the healthcare system, and if you do the math, it actually is a good investment to treat that person, cure that person, right? It's not more of a chronic, keep it down. No, if you can cure somebody with a gene therapy or a one-shot cell therapy, the economics actually do support the cost, and the insurance companies, are, reimbursing. Are they reimbursing for everything? Likely not. Which ones do they choose? Not sure, but at least they're doing it, and they're seeing tremendous, results. Imagine someone who is losing their sight. How much would you pay to get your sight back for your child? Right. So there's a give and take, but these are some of the most needy people needy in terms of disease, not finance, to be able to get these treatments and then be able to be part of the next study. So if you get the treatment, one thing that's nice is that you can participate in other follow-up studies, so they can see how that treatment, how does your disease progress, or stabilize, or improve. And so rare patients tend to be amongst the most generous in their time, because they want to make sure that someone else doesn't have that. Right. And so that ethical piece, that empathy, that these kids and adults, there's lots of rare diseases in adults that we work on, it's quite remarkable.
That's awesome. Yeah, and thank you for sharing your personal story, what got you into this, because it's come up a couple of times in our conversations. And my dad died 17 years ago, metastatic melanoma into his brain, and that's what got me into this originally. But my son had that rare disease, the DNET tumor, that luckily it was not life-threatening, and it was very treatable. And we had one of the best pediatric neurosurgeons in our backyard, so it worked out beautifully. And so we're very fortunate.
No, and sometimes it's just who you know. Right? So in this field, anybody that has a cancer diagnosis, they usually somehow call me. I'm not a physician, but I know many that are. And it's just, you just start fanning out. And sure enough, you get the top docs. Who are the top docs? You go on the internet, it is what it is, right? But if you get a personal recommendation for a physician, and that physician is willing to see you, you've now cut, again, months, if not weeks, if not years, to get to see them.
It makes all the difference.
And now part 2 with Chad Briscoe.
Russ, well, yeah, thanks for taking some time to chat with us today, and I had a few things I wanted to ask you about as well. As you were talking with Gregory, one of the things that came to my mind is what can we do in the pharmaceutical industry to help support this rare disease effort? Sometimes I feel like the patients are on their own, the companies that are developing rare diseases are on their own, and I don't feel like I've heard concerted efforts, let's say between CROs and the pharma partners, the pharma companies that are doing the rare disease research, to really figure out a better path forward to make this a little bit easier to get through clinical research. What have you seen in that space? Is there an opportunity to do more?
Yeah, there absolutely is. I mean, if you think about the challenges that we have and how can a CRO address those. Yeah. So there was a presentation today on how to deal with a very difficult biomarker to measure. Mm-hmm. And your site can do it because the phase 1 unit is there, mm-hmm. the flow cytometry lab is there, and you can walk that sample across. Yep. That sample cannot even be shaken, or it activates the biomarker. Yeah. So how do CROs set up more of these regional areas? So if you're one shop, it's not going to be able to be done. Right. But if you have units and labs mm-hmm. At those sites, now we can expand. Because what's happening now is we have to figure, do we want to do this biomarker at all sites? Well, if we need to do that, we have to train all these sites. And quite frankly, even the most simple assays are challenging. Yeah. Even if you've trained them, you can come back 6 months and they're not trained again. It's just muscle memory and all those things. So being able to collect, process samples and analyze them closer to the site, more the bed-to-bed side, or doing it at home with some patient-centric ways of collecting samples, by micro devices. And I think the other thing is to start to begin to understand where these rare patients live, meaning where should we build some of these units? Now, yes, they're spread out everywhere, mm-hmm. but are there universities or not-for-profits that really focus in this area? Mm-hmm. And can there be some sort of an alignment set up with them? Now, you're not going to develop a whole panel of rare disease biomarkers that are going to be used infrequently. Right. And so it's usually for a program, and that program will last 4 to 10 years, mm-hmm. Depending on what's going on. So the idea of really narrowing that time down. Time is our issue. So getting with these companies or these not-for-profits, working centrally, and in fact maybe setting up relationships. Right? It's we'd like to work with you. We have a whole group of people that want to work on myasthenia gravis, or that they want to work on a different disease. We have to go out and find all those KOLs and vendors. Right. So it may not be a bad idea to leverage. Here's what we need. Mm-hmm. We'd like to work on this, and this disease. Can you help us operationalize those studies? That's the hardest part. Finding the patients. Right, we spoke about how you diagnose patients and how long that takes. Well, is there another way of doing that by being closer with these investigators? Mm-hmm. For us, it's a challenge because there's so many investigators. But I think once you guys hook up, with a series, now you're the rare disease operational company. That would help us tremendously. I think that's where we really do struggle. And finding those patients, finding those clinical sites where you can train the staff. Not all staff are, at the bench at any point in their lives. And if you go in there and you're trying to teach them how to use a pipette, add antibodies, stain, and do other fixative type activities for flow, mm-hmm. it doesn't work. Or it works, and then all of a sudden it doesn't work. So things like the company providing training to the sites mm-hmm. versus the innovator doing that. Mm-hmm. You tend to have a staff that's definitely more fluid, mm-hmm. and globally they're all over the place, whereas we are located in one place. Yeah. So how do we leverage that so that we can focus on the analysis mm-hmm. Or developing the new biomarkers while at the same time you're laying out all this groundwork. It just takes so long. And the patients are few and far between. So the more of these sites that are identified, or at least cataloged. So I go to you and I say, hey, I got this disease, what do you have? In much the same way that you have sites that would focus on RA, or clinical sites that'll do neurotype disease. We need something like that we can just accelerate everything.
And do you think an organization like AAPS could be a conduit for some of those conversations? Is that maybe that's somewhere we need to go?
So you're seeing it, right? There's
well, you said there was a talk today, so that is
there's a number of rare diseases. So what has happened, right? So AAPS went from small molecules and antibodies, and now it has all these different modalities. Well, at the same time, there's rare diseases. There's a focus at AAPS on working in low and middle income countries, and I consider those diseases, even though they're large in number, they're rare in treatment. We can't get people to treat them properly. And so, for example, if you're looking at sickle cell disease, well, if you run those studies in the US, it takes a certain amount of time. If, however, you run those studies in Nigeria, you can probably do that study in 1/10 the time, because there's so many people with that disease. So even though it's, from a size standpoint, may not be a sickle cell, a rare disease, some people will think it absolutely is. So AAPS, between the global reach that they're doing, between the interest in working in novel diseases, which brings all those novel challenges. That's the big deal. We're learning new challenges. So the world has not gotten easier when it comes to drug development. You would think it does, and we have AI now, and that's supposed to cure the world. But it's only a part of what it is. And so things like AI, and there's a bunch of talks here on site selection using AI, on bioanalytical method validation, on reviewing method validation reports to try to find errors. All those pieces are feeding in. And for rare, it allows us just, again, to move a little bit faster in each one of those areas. And so AAPS is particularly this meeting. This meeting, for whatever reason, it pulled together rare, it pulled together gene therapy, the AAV. There must be a half a dozen talks on AAV, and we all have the same problem. Right? Anybody that's working in AAV has the exact same problem of dealing with pre-circulating antibodies, period. And so why not have a discussion here on how to harmonize those assays? Is there a way to have one assay or a diagnostic company that steps up to the plate so that I don't have to go get an IDE and another company has to get an IDE, or at least the assay is developed, and then you can take it to that IDE stage.
Do you think some of these I'm going to call them broad spectrum technologies, whether it's whole gene sequencing or like the Olink, is there going to be, when they're coming, I guess already, so I'm kind of answering my question, but are there, is that an approach to just say, we're just going to shotgun it and look for everything instead of trying to look for one thing or two things?
So without question, Olink has made proteomics easy. Right? It used to be very expensive pieces of mass spec equipment. Whether you're doing top-down, bottom-up proteomics, took a lot of resources and tremendous training. Not saying the Olink technology you can give to a child, but it is incredibly simple and robust. Now, those technologies 10 years ago, where they were measuring hundreds, if not thousands, of biomarkers, they had some issues, right? And you'd wonder, if your marker went up, good, but if your marker didn't go up, is it because the kit wasn't that good or the antibody wasn't that good? Whereas Olink seems to have solved a number of those issues, and it's now, I believe, likely in almost every one of our trials. Now, you take that, you get your big numbers, you'll see, and then you just down select and try to get it to maybe 2 or 3, and then you make your individual assays or your multiplex with 3. But absolutely, it's changed the ability to do biomarker discovery in a cost-effective way.
Oh, that's phenomenal. So Russ, 10 years down the road, what's the biggest change we're going to see in the pharmaceutical industry?
10 years down the road, what are we going to see? I believe companies are going to start to come together a little bit more.
Yeah, good. Optimism.
The gene therapy is the one I think we're all looking at. It seems to, you talk to some people and it's God's gift, and others, we can't do it. And again, I was on the, oh my God, how are we going to afford this, to seeing a gene replaced. I think it just happened this week where they restored vision to a child. Right, so I still think we can get there. I think on the cell therapy side, the sooner that we can move towards allogeneic cells versus autologous.
Autologous.
I'm going to say it again. The sooner we can get to
Autologous.
As soon as we can get.
We're not cutting that, by the way.
As soon as we can get to autologous, the better, right? So you got on-the-shelf therapies, that'll bring the costs down. That'll allow more people to be enrolled in studies and do those studies and actually conduct those studies at sites that aren't, waiting to deal with that sample that's coming in. Imagine it's the summertime and there's a hurricane that comes through, or the winter time and a blizzard comes through, and that sample that you're waiting, that they've taken from your body, they've augmented it, and now they're going to put it back in you, is stuck in customs, or it's stuck, on a tarmac someplace. So I think a lot of the logistics, it would be a big change on the infrastructure and how we ship things. But I think that as soon as we go to allogeneic cells, I think that's going to be the big change. And there's other technologies. There's I think it's in vivo CAR T, where instead of taking those cells out, augmenting them and changing them and putting back in, they're in the body and they're activated within the body.
Wow.
Right? So
Incredible.
boom, you don't have to deal with taking that out. So that's the part. And I think the last piece is what I call funky molecules.
Okay.
It seems that if you get the smartest protein chemist in a room, they can make molecules that can pretty much do anything. I mean, the idea of having an antibody, a bispecific with two different epitope binding regions, and now you see triple, and now you see T-cell engagers where we have one over here, we're going to bring these cells together. It's that sort of thing that nobody would ever thought about. Before it was just binding. Right? And now it's bringing things together, augmenting other cell types. And I think the last piece is likely going to be in the spatial area, where we can see exactly where that drug is. We, from a drug disposition, you want to know where it is. You can do this either by mass spec. There is, IHC, more imaging by pathology. But that whole field is growing. And then you layer on top of that the AI ability to read that and say, that's lung cancer, or that's lung cancer with this mutation because that's something we looked at. And I saw something just the other day where they can take images of your face, and through those images, and they have a whole battery of images, they can not only tell you what rare disease you have, but what the mutation may be. Because the face looks a little bit different with each one of those mutations. And imagine you just come to you come to a village or something, and you shine, a rather elaborate camera. It's not the big giant thing, and you can diagnose in the field.
Wow, that's cool. Fantastic, Russ. Thanks so much.
You got it.
Appreciate the time.
Anytime. Give me an huzzah. Huzzah. Huzzah.